tuning in to Planetterrian Daily, episode 123. Today is July 17th, 2026. Here's your science and health briefing. A new lab study shows many sweeteners alter gut bacteria growth, with some combinations sharply reducing diversity. Researchers tested 39 sweeteners and recorded more than 100 instances where the compounds changed microbial growth when mixed with medications, caffeine, or flavorings. The combination of isostaviol and the antidepressant duloxetine stood out for cutting beneficial bacteria and lowering overall diversity. The work shows sweeteners can act directly on microbes rather than only through host metabolism. Follow-up experiments will test whether these shifts appear in human gut communities. Now, shifting to a related molecular signal that influences how immune cells handle aging tissue. Blocking the prostaglandin EP2 receptor maintained the ability of tissue-resident macrophages to clear senescent cells in multiple mouse organs. The intervention limited mitochondrial decline and other age-related tissue changes. Restored clearance of senescent neutrophils was one measurable outcome. Researchers will next examine whether similar signaling changes occur in human aging tissues. A few episodes back, the show covered macrophage loss of clearance capacity with age. Today's blockade of this single lipid signal restored neutrophil removal and protected mitochondrial function across organs. Now, moving to another receptor-level approach that protects immune organ function with age. Targeting the GPR40 receptor preserved thymic structure and function in older animals, countering normal involution. The thymus produces T cells critical for immune defense. The aging cell study identifies a receptor-level intervention that could maintain immune capacity with age. Next steps include testing receptor agonists in additional models. On a different note, a living plastic material incorporates bacteria that trigger complete breakdown within 6 days once activated. The process leaves no microplastic fragments. The material targets single-use applications where rapid, clean degradation is needed. Further work will optimize bacterial strains and polymer compatibility for different use cases. From materials designed for controlled breakdown, next comes detectors engineered for cleaner performance in medical imaging. The Platon detector uses a light-producing material, a light field camera, and sensitive photon sensors plus AI to map particle trajectories. Simulations indicate performance equal to or better than current systems built from millions of separate components. The design simplifies scaling and could improve resolution in PET medical imaging. The team plans prototype construction and validation against existing detectors. Finnish players spend about €330 million yearly on golf, generating an estimated €630 million in total societal benefits, according to a Frontiers in Sports and Active Living analysis. The study quantified health, social, and economic returns beyond direct expenditures. Findings support continued public investment in accessible courses. A UN report outlines practical steps for governments to balance marine resource use with pollution control, climate adaptation, and biodiversity protection. It draws on the $2.2 trillion global ocean economy and the billions of people who rely on these ecosystems. Authors emphasize measurable targets for pollution reduction and habitat restoration. Austrian research found that water-related activities and access provide physical, social, and psychological benefits that extend beyond exercise alone. The study highlights how daily water interactions support mental health and community connection in later life. Results suggest urban planning should prioritize safe water access for aging populations. University of Rhode Island surveys located living freshwater mussel populations in the Moosup River for the first time in over 30 years. Several native species sensitive to water quality were present. Their return serves as a biological indicator of improving river conditions. University of Arizona tests showed graphene nanoribbons retain electronic properties after exposure levels relevant to fusion reactor environments. The nanoribbons could serve as durable sensors inside extreme radiation fields. Researchers will evaluate long-term stability under combined heat and radiation stress. A Nature Review's microbiology review synthesizes evidence that warming and altered precipitation change pathogen survival, transmission routes, and geographic ranges. Different pathogens respond variably, complicating uniform predictions. The analysis calls for region-specific surveillance and adaptation strategies. University of Seville researchers mapped microbial communities along the Guadalquivir River to detect contamination not captured by standard chemical tests. Shifts in bacterial composition flagged pollution sources and gradients. The approach offers a sensitive, low-cost monitoring tool for large river systems. Wildfire smoke reached unhealthy levels across the Midwest and East. The smoke came from distant fires. Visibility dropped and air quality alerts were issued from the Great Lakes to the Atlantic coast. Officials advised staying indoors during peak periods. MD Calc is introducing ratings for clinical calculators. The platform will score more than 800 tools. These tools are used for risk assessment, eligibility decisions, and diagnosis. Scoring will focus on bias and validity criteria. The system aims to help clinicians choose reliable calculators. Initial ratings will cover widely used scores. Plans call for ongoing updates. Axiom's Wharton's jelly-derived cells produced no deaths over 2 years in infants with severe neurological conditions. The Phase 1 data support the company's approach to targeted brain delivery. Further trials will test efficacy in larger groups. The EP2 receptor findings build directly on earlier work, tracking how tissue-resident macrophages lose clearance capacity with age. The result points to a concrete molecular handle on one driver of chronic inflammation. Human tissue studies will be the clearest next indicator of whether the pathway behaves similarly. Most people assume non-caloric sweeteners pass through the body without affecting anything except taste receptors. In reality, they reach the colon and interact directly with resident microbes. Right now, as you digest a diet drink, certain bacterial species are already adjusting their growth rates in response to those compounds. One lab survey found over 100 distinct interaction patterns when sweeteners were combined with common drugs or flavorings, including sharp drops in microbial diversity. A single memorable example is the pairing of isostaviol with duloxetine, which cut beneficial populations more than either substance alone. The practical takeaway is to notice how your own digestion or bowel patterns shift when you change sweetener intake, and to watch for larger human feeding studies that will test whether these lab shifts translate to measurable microbiome changes. New tools for tracking cellular aging and environmental exposures continue to refine how we measure daily health influences. Keep an eye on upcoming human tissue studies that will test whether the EP2 pathway behaves similarly in people. That's Planetterrian Daily for today. If you enjoyed this, a rating or review on Apple Podcasts or Spotify really helps new listeners find the show. I'm Patrick in Vancouver. Thanks for listening, and I'll see you tomorrow. And if you'd rather watch than listen, find us on YouTube at Nera Network. Links in the show notes. One more thing, if you liked today's episode, our sister show OmniView is worth a spot in your feed. A balanced daily world news briefing that takes every side seriously. It's one of the Nera Network's daily briefings, all free at neranetwork.com. The Do Positive Pod is the club for people who do something about it. Episodes and the dispatch wall at neranetwork.com/thedaypod. This episode used AI voice synthesis of my voice. Editorial selection and analysis are my own.